Welcome to the stem cells module. In this talk, we're going to learn about the pluripotency that is observed in stem cells. A stem cell is a type of cell that has the ability to divide and produce copies of itself, which is what is shown in the figure, and this is called a self-renewal. Now, a stem cell can also undergo differentiation where it is able to generate progenitor cells. And these progenitor cells will then have the capability to form different cell types. And this is called as potency when a cell is able to form different types of cells through the process of differentiation. We're now going to look at the different types of stem cells based on their potency. Now, the stem cell potential is the range of cell types a stem cell can produce. And the first type of stem cell that we're going to look at is a totipotent cell. A totipotent stem cell is a stem cell that can generate all cell types of both the embryonic and extraembryonic lineages. When we look at the blue box, the cell that is shown to be totipotent is a zygote. So a zygote is a totipotent cell because it has the ability to give rise to embryonic as well as extraembryonic cells. When the zygote undergoes cell division in the cleavage step of development, the cells up till the 4 to 8 cell stage are also totipotent. The next type of stem cell is a pluripotent stem cell. This is a stem cell that is capable of producing all cells of the embryo. However, it cannot produce the extraembryonic lineages. Another type of stem cell is a multipotent stem cell. Multipotent stem cells can produce cell types with restricted specificity depending on the tissue or the organ they are present in. Thus, they are not as versatile as the pluripotent stem cell. So an example is a stem cell that is present, let's say in the brain, can give rise to cells that are present in the brains like neurons. However, that stem cell will not be able to give rise to, for example, a stomach cell or a liver cell. Before we go to look at the different types of stem cells and their niches, let us first look at early embryonic development. Now, during early embryonic development, after fertilization, we obtain a zygote that is shown in the blue box in the figure. The zygote can undergo cell division to form a 2-cell stage, then a 4-cell stage, and finally it forms a cluster of cells that we call as a morula. The morula then will undergo further development to form a fluid-filled cavity which is called as a blastocele. Now this embryonic structure that has the blastocele formed, it develops into what's called as a blastocyst. In the blastocele stage, the outer cells that are present are called as the trophectoderm, while the cells inside, which could be approximately 12 cells, are called as the inner cell mass or ICM. This is what we can see in the figure shown in the blue box. The cells that are light blue that form the periphery of the circle are the trophectoderm, whereas the cells that are inside the circle are the inner cell mass. The inner cell mass cells are developed to form the primitive endoderm and an epiblast. So as shown in the figure, we can see that the inner mass cells undergo changes to form either a primitive endoderm that is shown in the yellow, or they become the epiblast. Now, the cells that are present in the epiblast will actually form the embryo, while the other cells, namely the trophectoderm and the primitive endoderm, they proceed to form the extraembryonic structures. These extraembryonic structures can be in the form of placenta, or it can also be in the form of the yolk sac that's present in the embryo. The embryonic stem cells, which are pluripotent, are derived from the ICM or the epiblast. So the are the network of cells that are formed before we have the epiblast and the primitive endoderm. So the inner cell mass as well as the epiblast that is formed later in development both have pluripotent capabilities. We're now not going to look at mechanisms for promoting pluripotency of the inner cell mass cells. Now, when we look at the formation of the inner cell mass cells, they arise due to asymmetric localization of factors, which usually occurs in the outer cells of the morula. This is before the inner cell mass has been accomplished. Cell division perpendicular to the apicobasal axis, which is the apical to basal or the outer side of the embryo to the inside axis, results in daughter cells, one of which becomes an ICM cell. So when we look at the figure here, we can see the cells that are present on the ring, which are the trophectoderm cells, and the apicobasal axis is shown in the blue line, which is running from the outer side of the embryo to the inside of it. Now, if we have cell division perpendicular to this axis, which is what is shown in the form of the red line in the figure, then what will happen is cell division happens through this perpendicular axis as shown in the middle figure. So in the middle figure, we're able to see one of the cell undergoing cell division. And in this case, what will happen is we end up with two cells. The first cell being a cell that remains as a trophectoderm, and the second cell turns out to be the inner cell mass cell. This is the cell that is to the interior of the embryo. Now, what happens if we have cell division parallel to the apicobasal axis? So when we look at the figure and look at the part that's in the box, in this case, the cell is undergoing cell division at an axis that is parallel to the apicobasal axis. So here we can see the red line showing the axis of cell division, which is parallel to that apicobasal axis that we saw in the previous slide. When such cell division occurs, you end up with two daughter cells, but neither of these cells will be the inner cell mass. Both of them will actually end up being the trophectoderm cells, and this is a way in which we can expand the trophectoderm that is present in the embryo. To understand how the ICM cells are formed, let us look at a pathway that plays an important role, which is the Hippo pathway. Hippo is a kinase that can be activated by different ligands and receptors. Now in the figure, we're looking at the Hippo pathway in Drosophila and in mammals, and the pathway basically works the same way, but the members involved in the pathway have different names in the Drosophila system versus the mammalian system. Now let's first look at what happens in the absence of Hippo activation. When Hippo is not activated, the transcription factor called Yorkie in Drosophila is able to activate the transcription of target genes. In the case of mammals, the equivalent of Yorkie is YAPTAZ. And so, in the case where Hippo is not active, YAPTAZ in the mammalian systems is able to activate target gene transcription. Now let's see what happens when Hippo is activated. So in the case of Drosophila, when Hippo gets activated, Hippo is able to activate its kinase function. Now what we're showing here in the blue box is Hippo and Drosophila, and now we're looking at the second blue box where we have the mammalian equivalent of Hippo. In both cases, activation of the kinase is usually through phosphorylation. And once the kinase gets activated, it is able to activate or phosphorylate its downstream protein targets. These protein targets will ultimately phosphorylate the Yorkie protein in the case of Drosophila, or the YAPTAZ protein that's present in the mammalian system. Phosphorylation of either Yorkie or YAPTAZ promotes its cytoplasmic retention or in some cases can mediate the degradation, especially in the case of mammalian systems. And this way, the cytoplasmic retention or degradation of Yorkie or YAPTAZ does not allow these proteins to activate gene transcription. And this is how the Hippo pathway works. So now let us look at how this pathway is used in the case of ICM cell production. In order to understand how the inner cell mast cell is formed from the cell division of the trophectoderm cell, what we know is that the differential localization of specific proteins within the cells leads to activation of different gene regulatory networks, and this causes the acquisition of different cell fates. So, when we look at the trophectoderm cell, which is shown in the blue box, the trophectoderm cell, the upper part is the apical part of the cell and the lower part is the basal part of the cell. Now let us see what happens in the red box. Now in the red box that's shown in the figure, we can see that there is homophilic binding of E-cadherins that are present on the cell membrane of the trophectoderm cell as well as the ICM cell, which is shown in pink. Now on the part of the ICM side, we can see that the E-cadherin is interacting with the protein that is called angiomotin, also shown as AMOT or AMOT in this figure. This interaction is happening in the basal part of the trophectoderm cell. Now let us look at the apical part of the trophectoderm cell. In the apical part, we don't see the expression of E-cadherin. So there is differential localization of E-cadherin. It is present mostly in the basal part and maybe to a certain extent on the lateral sides, but we don't see E-cadherin present on the apical part of the trophectoderm cell. We do see angiomotin, but the activity of angiomotin is inhibited through the presence of these apical proteins that is shown in the figure, which are basically PAC and APKC. Now let's look at what happened. Now, because of the interaction of AMOT with the apical proteins and not with E-cadherin, the hippokinase pathway does not get activated. Since the hippo pathway is inhibited, YAPTAZ is able to bind and promote the transcription of genes, which in this case is CDX2. CDX2 gene is able to provide the signals for the cell to remain as a trophectoderm cell. Now let us look at what happens in the ICM cell, or the cell that is adjacent to the trophectoderm on the inner side of the embryo. So this cell is what is shown in the blue box, and I had explained before that in this cell we can see AMOT, which is shown as that C-shaped orange colored structure, is able to interact with E-cadherin. This activates the hippo pathway, and so the interaction of AMOT with E-cadherin results in the phosphorylation of hippo and the cascade begins. That is what is shown in the red box. Once the hippo cascade is initiated, what we can see is that YAPTAZ is going to be degraded or retained in the cytoplasm. And since YAPTAZ is now not allowed to stay in the nucleus, CDX2 expression does not occur in the ICM cell. What we can also see is there are other transcription factors that are able to make the cell express the protein OCT4. OCT4 is a protein that plays an important role to activate the pluripotent gene regulatory networks. Thus, the main difference between the trophectoderm and the ICM cell is the presence or absence of CDX2. Additionally, in ICM cells, we are starting to see the expression of genes that are important in maintaining pluripotency. With this, we come to the end of our talk, where we learned about potency in stem cells and the different cells that are formed during the early stages of development. We also learned about the Hippo pathway and how the Hippo pathway is able to promote pluripotency in embryonic cells.